My name is Margot Pont. I'm a VP of Translational Development at Integra Therapeutics, a Barcelona-based company founded in 2021. And I've recently joined Integra back in September of last year. I have a long experience in leading both biomarker strategies and translational research, both for CAR-Ts and TCR-Ts. And what I've really learned when that field started to revolutionize and started to be taken up is that it's really a new modality. So the original thinking of how pharma was operating, being used to small molecules or then later with biologics, is that as CAR-T cells or these TCR-T cells are a living drug, the field doesn't really fit the way things are organized, the way people are thinking about it. So if you think about biomarkers or clinical pharmacology, these things aren't silos if you're talking about a living drug. They're all interconnected, and to me, it's all T-cell biology. So we really need to think about it differently. And luckily, the regulators, also because of the significant clinical breakthroughs that we've seen with these therapies, luckily the regulators have seen that possibility and have really worked to allow for innovation on that side. But I think the larger industry also really needs to adopt this new way of thinking. And while the field moves along and new innovations come, that we need to continue to do that. FiCAT is a groundbreaking gene writing platform where we really combine the specificity of a nuclease together with the transposase to get the cut and integration efficiency. And with that, we can deliver large gene payloads at a site-specific manner. So really allowing fine-tuning and site-specific integration with the possibility of delivering large gene payloads instead of the very small gene edits that are sometimes made by others. And I really think that opens up so many different possibilities that go beyond the current standard modalities such as AAVs or LVVs, where payload size is really one of the limitations. And so with this, you can do more simple, complex editing, but in a much more simple way, just in one step doing knockout and delivering a large gene payload at the same time. With giving a larger payload design, this opens up a whole venue of new possibilities. You can include a kill switch, you can include markers to select and enrich for the cells that you want. Yesterday also in the panel, we discussed the option or the necessity of having a kill switch in there. And this gives you control, again, of this living drug that you infuse into a patient. Once you infuse it, it can go off and do its thing. So if you introduce a kill switch, this will give physicians control over that living drug later on in the patient if something would occur that would want to stop the treatment. Or you could also use it to actually avoid maybe exhaustion of the cells while it's encoding antigen. So you can turn it off temporarily and then possibly also then turn it back on to then exert its function again. So really fine-tuning what this living drug can do in a patient. Thinking about how to translate this from in vitro to then in vivo, but really beyond that to go towards the clinic. I think the challenges is that intermediate step of how to find good in vivo models, and are in vivo models really what you would need to be looking at. So for each indication or specific question, can you find a model that is really predictive of what you would see in the end in clinical use? And for that also, there's now a push for new alternative methods and really actually having in vitro models and reducing the use of in vivo models and really finding good technologies to have good in vitro predictable assays, to move away from that, to have something that would really fit and predict what would happen clinically. If you ask me what type of innovation I'm particularly excited about, I'm truly a T-cell biologist, really from training and all of my career. So for me, having these complex gene-edited T cells that can target tumors or other indications and really make a difference there. But I think we shouldn't focus just on one T cell, on one subset or one thing alone. I think it's combinatorial. So having engineered T cells that can recruit other populations of the immune system that can then come in and help them do the job. We shouldn't focus just on the one population. So that's something that really excites me. Yeah, so Integra is developing its first rare disease in a liver indication, gene therapy. And we can do that because of the FiCAT technology that we have. And we feel very lucky to be supported by an EAC accelerator grant to develop this further. And I think that's one of the key challenges is in a rare disease population, trying to find the funding, trying to find the right target patient population, but also then really bringing that therapy further to make sure it can reach all the patients that it needs. So we feel very lucky to have at least this EIC Accelerator grant. We feel encouraged by the recent regulatory steps, both by FDA and also in Japan, when there's a clear focus on further development of rare disease indications. And we're always open also to further partnering opportunities to develop this further. From my perspective, AI can accelerate the field significantly. And we're using AI at Integra to also do novel protein design. And last October, we published a Nature Biotechnology paper together with our partners of University Pompeu Fabra and the Center for Genomic Design in Barcelona, where we really use AI to change and to predict new proteins. And that has led to improved transposases to then further also optimize our FiCAT platform. So we're very excited about the possibilities that AI can bring for new protein design. So the biology is no longer the limitation. You can have AI really help you design new biology. I think some of the challenges to make these therapies more affordable and accessible worldwide is trained personnel, being able to have the right people to actually develop these therapies. A lot of them are still very manual process. More and more work is being done to optimize that, but you still need operators that are skilled and qualified to do the work. And with this acceleration, we see a lack of highly trained personnel that's required. The same is true. So infrastructure, logistics, that comes with cost of goods. So trying to come up with ways to reduce cost and to really give this access to patients globally will be a very important step to take.